In this video, we will learn about the reproduction and development. We will focus on the sexual reproduction in flowering plants. First, we will look into the reproductive organ of the plant, which is the flower. The flower consists of 4 types of modified leaves: the sepals, petals, stamens, and carpels. Next is the exhibition of a few diagrams that consist of beautiful and colorful flowers that designed to attract pollinators. We have this unique flower like this that appealing, and a huge flower like this rafflesia that deserve our attention. The parts of the flower consist of stamen, which represent the male reproductive organ, the ovule, which represent the female reproductive organ, petals, bright and colorful to attract pollinators, and the sepals to protect and support the flower buds. Now we will focus on the male gamete. The male reproductive organ consists of microsporangium or pollen sac in the anther of a stamen. Inside the microsporangium, we have the microsporocyte, or also known as microspore mother cell, divide by meiosis to produce haploid microspore. A cluster of 4 microspore is called tetrad. Each microspore undergo mitotic division to develop into a pollen grain, which, protected by pollen wall, consists of exine and intine that will protect the male gametophyte. The male gametophyte then undergo mitotic division to form generative cell, which later on divide to form 2 sperms, and the tube cell will produce pollen tube that penetrates the style after pollination takes place. Next, we will look into the female gamete. Female gamete begin with megasporangium, that also known as ovule. Inside the megasporangium, the megasporocyte or megaspore mother cell undergo meiosis to produce 4 haploid megaspore. 3 megaspore degenerate and only 1 survive to form the female gametophyte. Before it turns into female gamete, the surviving haploid megaspore undergo 3 times mitosis without cytokinesis to form cell with 8 haploid nuclei. Now, the ovule, which contain 3 antipodal cells, 2 polar nuclei, 2 synergid cells, and 1 egg, now will be called embryo sac. The 3 antipodal cells at the callosal end of the embryo sac with unknown function. Polar nuclei, when fertilized, will form the endosperm. The egg cell, when fertilized, will form the zygote. Synergid cell will attract and guide the pollen tube into embryo sac. We further discuss the development of male gametes. It begins within the pollen sac of the anther, in which the microsporocyte or microspore mother cell divide by meiosis to produce 4 haploid microspore. Each of the microspore divide by mitosis to form generative cell and tube cell. Generative cell undergo mitosis to produce two sperms. The tube cell divide and differentiate into the pollen tube that delivers the sperm to the egg and embryo sac. This is the pollen grain, which protects and encloses the immature male gametophyte. Next, we will further understand the development of female gamete. It begins within the ovule, which consists of a diploid megasporocyte. The megasporocyte, or also known as megaspore mother cell, undergo meiosis to produce 4 haploid megaspore. Only one of the megaspore survive, while the other 3 become degenerated. The surviving megaspore undergo 3 times mitosis without cytokinesis to form embryo sac with 8 haploid nuclei, consists of 3 antipodal cells, 2 polar nuclei, 2 synergid cells, and 1 egg. Now, we will discuss about double fertilization. Double fertilization is the union of 2 male gametes with 2 different nuclei within the embryo sac. One male gamete will fertilize the egg to form zygote. Another one male gamete will fuse with 2 polar nuclei to form triploid nucleus that will form the endosperm. After landing on the stigma, the pollen grain will germinate, in which the pollen tube will extend down the style and migrate towards the ovary. Generative cells undergo mitosis to produce 2 sperms. Pollen tube will enter the ovary through micropyle to discharge 2 male gametes into the embryo sac. Within the embryo sac, 1 sperm will fuse with the egg to form zygote that later on will develop into an embryo. Another male gamete will fuse with 2 polar nuclei to form triploid nucleus that will form the endosperm, the food storing tissue of the seed. After fertilization, the ovules will develop into a seed. The ovary develop into a fruit that encloses the seed. In angiosperm, double fertilization will ensure the endosperm only develops within the ovule when the egg has been fertilized. This will prevent the wasting of nutrients in eggs that has no embryo.